feeling behind my eyes. You stood out 'cause you were in red. Then, against my intentions, I let you turn me violet instead. Summer high, met you in August. New York, back to my cold apartment. Distance sucks, but at least I'm still with you. White out skies, middle November. You and I, flights to wherever. I don't mind, as long as it's with you. As long as it's with you. The festival that we met at, where you were just a face in a crowd, and now I can't imagine me with anybody else. As we close out the night underneath the lilac sky. Summer high, met you in August. New York, back to my cold apartment. Distance sucks, but at least I'm still with you. White out skies, middle November. You and I, flights to wherever. I don't mind, as long as it's with you. As long as it's with you. As long as...
Olá, pessoal, tudo bem? Boa tarde, bem-vindos e bem-vindas aí a mais um momento da SEB, Semana da Engenharia Biomédica do Inatel. É um grandiosíssimo prazer ter vocês aqui e a gente está nesse momento, um momento muito especial para a gente. Durante esse momento que a gente está vivendo aqui agora, são várias comemorações, tanto da comemoração da engenharia biomédica por si. Essa é uma carreira muito bonita, uma carreira que consegue modificar e melhorar vidas, né? E tem esse impacto gigante na vida das pessoas. E também a celebração dos 10 anos do eHealth, o Centro de Desenvolvimento de Tecnologias para a Área Médica. Mas sem muitas delongas, eu já vou começar a apresentação com vocês. Antes, gostaria de agradecer imensamente aos patrocinadores do evento, a empresa Equipacare, Master Medical e Biotron, que graças a vocês a gente tem conseguido fazer várias dinâmicas e vários momentos especiais aqui durante essa semana. Nesse presente momento, a gente vai ter uma palestra muito especial, que é de ela foi minha orientadora e também minha amiga hoje, Emily Dosmar. Ela é uma engenheira biomédica dos Estados Unidos, ela inclusive fez o pós-doutorado dela também em engenharia biomédica. E ela vai conversar um pouquinho com vocês agora sobre nanotecnologia. E também vai dar algumas dicas e algumas ideias sobre como que é o processo de educação e ciência lá nos Estados Unidos. Então, quem tem vontade de bater asas e viajar pelo mundo aí, vale a pena ficar bastante atento às dicas que ela for dar sobre essa cultura de pesquisa e de ciência que a gente tem aqui. Tá bom? Perfeito, o pessoal está comentando aqui comigo uma coisa muito legal. Para quem não fala inglês, nós temos uma live, fica aqui embaixo, né? Na descrição ali. A gente tem um link para quem quiser acessar aquele link e não fala inglês, a gente vai ter uma tradutora. E aqui, muito obrigado à equipe de tradução, né? Que eles vão poder fazer a tradução para vocês da palestra, em tempo real. E para quem quiser se arriscar no inglês, já manja bastante do inglês ali, vocês podem continuar nessa chamada, nesse link, porque é aqui onde a gente vai ter a palestra feita pela Emily na língua inglesa. Tá bom? Então, a gente tem esses dois links, esses dois canais, para vocês poderem aproveitar bastante esse momento aí. Combinado? A partir de agora, então, eu vou chamar aqui para o palco, nosso palco virtual, a Emily, Emma, né, como eu costumo chamá-la, para que ela possa conhecê-los um pouco aí. Ok? Então, a partir desse momento, eu vou virar a chavinha para o português. Hey, Emma, how are you doing? I'm very, very happy for having you here with us this afternoon here in Brazil, and also I think afternoon there in the States. I'm very, very glad for this moment with you and for having your time to give your presentation and sharing your experience with the guys in this very special moment for us. Thank you so much, Emma. Feel free to introduce yourself and start your presentation.
Thank you so much for having me. This is very exciting to be here today. Hello, all the way from, I'm in Wisconsin right now on the lake with my family. So I'm sorry if the internet is a little bit weak. So as Felipe said, I am a professor of biology and biomedical engineering in the United States in a school called Rose-Hulman Institute of Technology in Terre Haute, Indiana. And in addition to my research, I'm going to talk about graduate experience in the United States. So going to graduate school and what that is like, and what led me from undergraduate to being a professor today. So if you don't mind going to my next slide.
Okay, Emma. Just a second. Can you see your slides?
Yes.
Okay, great.
Yes, so I completed my undergraduate degree, my bachelor's of engineering, at Rose-Hulman, where I now teach. It's a very small STEM school, so we only offer majors in science and technology and engineering and math. It's located in Indiana, which is in the Midwestern part of the United States. And it's a lovely little campus. One of the things about Rose-Hulman is that it is not a PhD granting institution. So you cannot earn a PhD at the school. And so as a result, I was able to have a lot of research experiences as an undergraduate that I may not have been able to have at a larger school. When I was about a junior in college, I took a class in material science, and I fell in love with material science and was very, very excited about the topic. And I asked the professor if I could do research in his lab. And I spent the next year and a half working on a project in his lab, making very thin films out of a polymer that we could do mechanical testing on. And I really enjoyed the research experience, and so I decided I wanted to earn a PhD. And so I started looking around to see what graduate schools I might attend. And so if you're an undergraduate and you are thinking about attending graduate school, this next part is for you. So if you don't mind, yes. So one of the first challenges that you are tasked with when looking around to join a graduate department is to pick a department and a lab to join. And I have this quote up here: every project is a little bit interesting and a lot boring, because I see a tendency among undergraduates to want to pick the coolest sounding project at the most prestigious school. And it's great to go to a top school, and it's very important that you're interested in the research that's happening. But you should also consider the fact that even if a project sounds very cool, the day-to-day of research can be a little bit boring and tedious. So at the end of the day, you're going to spend many hours in the lab measuring things over and over and over again, waiting for tests to run, measuring and sampling, and it can be a little bit tedious. And so this picture I drew of myself, I doodled it in my lab book. I was waiting for a centrifuge to run, and I was imagining myself doing something more exciting. And so it's important, because graduate school is very long, that you are also happy as a person. And so if you are someone who loves to hike, you might pick a school near mountains. Or if you're someone who loves a big city, you might try to go to school in a big city. And so it's okay to prioritize things that will make you happy as a person, because those are things that will make you a productive graduate student. And next. Other things that you should consider when picking a graduate school are, does the department that you're joining have a qualifying exam? Now, at the end of my first year of my PhD program, I had to sit for a written exam that covered math, physiology, and then we had to read a paper and write a summary and present that to a committee of faculty. And so find out if the school that you're joining has a qualifying exam, and find out what happens if you do not pass that qualifying exam. So if you don't pass, does that mean you are kicked out of the program? You retake it. Look into the funding. So is the department well funded? Is the faculty who runs the lab that you want to join, do they have a grant? How will you be paid? Because as a PhD student, you typically are paid for your work and you do not have to pay tuition. Look into the cultural differences between the different schools that you are considering. The way to find out about some of the cultural differences are to contact graduate students who are in the department and ask them, how are the labs managed? Is your advisor someone who you can come in contact with regularly, or do you have to ask them for a meeting? Do they step into the lab? Ask them about their managerial style. because at the end of the day, the PhD, and the joke is that it's a little bit like a marriage. You are connected to your advisor for at minimum 5 years, but it can be as many as 7, and then extended beyond that if you continue to collaborate with them. And you want to make sure that you get along and that there's a good personality fit. And so do your research about what lab you'd be joining to make sure that it's right for you. Next. Another thing to look into is to consider your academic genealogy. So I completed my undergraduate degree at Rose-Hulman, and while I was at Rose-Hulman, I had this advisor, Dr. Jamil Ahmed, who you can see right above me. He had completed his PhD under Dr. Rob Linsenmeyer at Northwestern University, and in his lab as a graduate student was Dr. Jennifer King-Mueller. I was interested in doing research for her, and so when I knew that they were connected, I was able to ask for a letter of recommendation directly from someone who knew me and knew her, and that helped me get accepted into her lab. And so if you're an undergraduate and you know Felipe here, he is your academic genealogy. He knows me, right? So those are connections. Figure out what connections you have and use them to your advantage. Use them to get an introduction. And also don't burn any bridges, because these are your recommenders. And so it's a good resource to getting connected to the school and the research that you want to do. Next. So for me, I was really interested in Dr. King-Mueller's lab, who I think you'll be hearing from her later on in this week. And I wanted to be in a big city. I'm from Chicago, and being in Chicago was really important to me. And so I joined her lab at the Illinois Institute of Technology in Chicago. Next. A couple other notes about graduate school, in case you're wondering. The average time for a PhD is about 5 to 7 years, and the beginning part of that are taking classes. So you'll take about 12 classes, approximately a couple years worth of classes. At the end of some amount of time, for me it was a year, in other programs it's longer, you take a qualifying or a preliminary exam where you demonstrate competency in basic areas of engineering or whatever subject you're studying. And then you may have to serve as a teaching assistant. I did. A little later on in your graduate school experience, you'll need to take a comprehensive exam where you propose what you're going to do for your thesis to your committee. So you say, this is the research that I'm going to do. You do all that research, and then eventually you will defend that research to your committee, and they decide if you're ready to graduate. Next. So here is a picture of Dr. King-Mueller's lab, 2 pictures actually. At any given time, we had between 2 and 4 graduate students in the lab. Dr. King-Mueller is there on the right in her red coat, or in her red dress. And the other picture is when we got to meet Dr. Robert Langer, who is sort of the father of tissue engineering. That was a very exciting day when we got to meet him. And we had undergrads join our lab throughout the time that I was there as well. And the research happening in Dr. King-Mueller's lab had to do with the eye. And that's mostly what I'm going to talk about today. So she was looking into a lot of different things regarding the eye, but mostly she was curious about ocular drug delivery. Next. And so here you can see a couple of different methods for how you might get drugs into the eye. The thing that you're probably most familiar with is the idea of eye drops. Most people have dispensed eye drops into their eyes, and you know that it's something you can do at home yourself, but you also know that when you put eye drops into your eye, oftentimes tears run down your face and you lose a lot of the dose. And so that is a big challenge of eye drops. And also, even if you get the drug into your eye, if you get the drop into your eye, there are many barriers, tissue barriers, between the surface of the eye and the back of the eye that the drug must penetrate. And so if you're trying to deliver a drug to the very back of the eye drops are a very limiting approach, because you end up losing 95% of the drug to various tissue layers. And so there are alternatives to this. You can inject drug into various locations in the eye, and it can better travel to the back of the eye. On the next slide, I'm going to show you a picture of an inflamed eye, so just be forewarned. Yeah. So the condition I was looking to treat and prevent is called endophthalmitis. And this is an inflammatory condition of the ocular cavity, mostly of the surface of the eye. And it's the when microflora that live on the surface of the eye and on the skin are able to gain entry into the eye during either a surgery or during a trauma. And while it's very rare, it can be very catastrophic and lead to, I think 95% of the cases will include either reduced vision or blindness for the patient. And you really want to prevent it because it can only be detected after it becomes an event. And so it's really important that during surgery, a sterile environment is maintained and we prevent this infection from occurring. And so when you're having an eye surgery, the way you avoid introducing endophthalmitis to the patient or introducing bacteria to the patient's eye is next. The current approach is a bolus injection of an antibiotic drug at the conclusion of a surgery, and then the patient will follow it up with 1 to 2 weeks of antibiotic drops. And I already talked about some of the limitations of eye drops, but the bolus injection also has some limitations because the typical location where this drug is injected is into the vitreous. But if you inject into the vitreous, there is a small risk of retinal detachment. And so it's not an ideal place for injection. Next. And so some surgeons have started to inject the drug into the subconjunctival space, and just you can see in the image where that is. And it has some advantages over the intravitreal injection because there's a lower risk of retinal detachment. So it's a very safe location to inject. And the permeability to drug is much better than if you administer the drug topically. And so in theory, it's a really great place to inject a drug because it's safe and the drug can penetrate through the tissue. Next. In some early studies, researchers injected antibiotics into the eyes of both rats and patients, and they looked to see how long and how much of a peak of drug they could measure in the vitreous following subconjunctival injection. And what they saw was it took about 5 hours in both studies to peak the amount of drug in that location, and then very rapidly that drug is cleared from the eye, probably due to the aqueous humor turnover that is clearing the drug from the eye. And so a bolus injection is still not the best opportunity to give a drug to the vitreous because you get a lot of drug clearance and it doesn't maintain an effective dose for a long enough period of time. Next. So when I was looking into a project, I hypothesized that I could use a controlled extended release subconjunctival implant that was loaded with antibiotics, and I would be able to maintain a therapeutic dose of drug for a longer period of time in the vitreous that would sort of outperform a bolus injection. So because we have a lot of undergraduates in the audience, I want to just take a moment, next thing, to talk about why you might choose a sustained release drug delivery system over a traditional drug delivery system. And this is true not just in the eye, but anywhere in the body. So with conventional drug formulations where you just take a dose of drug or you receive an injection of a drug, you have these peaks and valleys associated with drug dosing. So when you first receive the drug, you have a large amount of drug in the system, and then that gets cleared away, and that drug will dip very low. And you might think about when you've taken, for example, a painkiller like an ibuprofen or an aspirin. If you're in pain and you take a pill, the pain goes away, but at the end of that window of when the dose is meaningful, so 4 to 6 hours, at the end of that, you start to be in pain again. And then you have to say, do I take another pill and potentially have too much drug in my system? Or do I have to wait and be in pain for a little bit until I can take the next drug? So the idea behind sustained release is it sort of maintains that steady level of drug for a long period of time, so there aren't these peaks and valleys in the drug in your system. Go ahead, next. And so here's another image of that. Just there are different, depending on the drug and depending on the sustained release system, you can see how the drug concentration may change in your system over time. Okay, go ahead, next. So I propose to build a sustained release drug delivery system out of polymer-based hydrogels. And I wanted this drug to be injectable into the subconjunctival space, filled with drug that could maintain a therapeutic concentration of antibiotics for 10 days. Now, go ahead to the next. I mentioned before that when you're in graduate school, you have a committee. So this is made up of a bunch of faculty members who are basically your peer review for your research. So they assess if you've done enough work and if your work is meaningful to allow you to graduate. And part of the process of graduate school is telling them what your plan is for graduate school for your research, and they give you some feedback and they help you decide if your project is realistic and if it will make an impact on the field. And so this is what I told my committee that I was going to do. Next. I said that I was going to develop a system that provided a steady and consistent dose of antibiotics for 2 weeks. I wanted to maximize the amount of drug that reached the target tissues while minimizing the drug lost. And I would completely eliminate the need for patient-administered topical drugs. And I said, next, I said my project would have 3 aims. This is very typical in a graduate project. You develop 3 primary aims for your project. I said I would develop that system in vitro. I would then test it in vivo. So I would look into a rodent model and I would test that system out to see how it performed in an animal model. And then I would model that system in MATLAB using programming to see if I could model how much drug I needed to load it with to optimize it. Next. So my first goal was to develop an injectable drug delivery system for the antibiotic vancomycin that I was looking into. Next. So I mentioned that I wanted to make my system out of hydrogels. If you're not familiar with hydrogels are typically made up of polymers. Polymers are very long chains of monomers. So they're a very long molecule made up of little monomers repeated over and over and over again. And you can introduce chemical bonding across those chains. Usually these are chemical bonds, sometimes they are physical bonds, but we won't talk about that right now. You can induce this bonding a lot of different ways and it create a 3D network. That 3D network will then be able to take in water and swell with water, which is why they're called hydrogels. And hydrogels have a lot of advantages because they can be loaded with drug and allow for that drug to be released for an extended period of time. And you can tune the mechanical and physical properties of a hydrogel by changing certain chemical parameters. You can make them injectable and you can make them biodegradable. Next. So here's just one small example. If you want to change the size of the network, you can do that by introducing more cross-linking, and that will have an effect on the property of that hydrogel. So you can increase the elastic modulus, the compressive modulus, and the stiffness, but that in turn reduces the amount of permeability or swelling of that hydrogel, and vice versa. And so you have a lot of control over the physical properties, but it gets complicated because if you change a polymer that will form those little cross-links in the presence of visible light. I looked at another system where I loaded little nanoparticles into the hydrogels, and I looked at a third hydrogel alone that was thermoresponsive. Next. So those visible light hydrogels, let me say one more thing. Okay, so the other thing is when we look at these hydrogels and we look at any sustained release drug delivery system, typically the release looks something like this. So this is sort of a standard release curve. And what you see here is drug over time. And on the left side, you see the percent of drug released. On the right side, you see the actual amount drug released, but it's just amount of drug over time. And this is cumulative, right? So we're adding at each time point the amount of drug that was released. In the beginning, you tend to see a rapid amount of release. We call that the initial burst. And then after some time, this plateaus. So I'm going to refer to the initial burst of a drug delivery system, and that's what I mean is how much is burst in the first 24 or so hours. Okay, next. So the visible light hydrogels that I mentioned, I evaluated for release and degradation and ability to load the drug in. And what I found was I was able to make a really tight mesh so I could load the drug in. They degraded in less than 14 days, so that was nice for clearing. And I was able to manipulate some of the properties. So I was able to figure out what I needed to do to change the stiffness or the elasticity of the hydrogel. But there were some problems with it. I couldn't make them injectable because as I made them stiff enough to be held together and to hold drug, they got so brittle that they fell apart. Because they degraded so quickly, all of the drug released immediately, and so I couldn't get them to release for 10 days. And I couldn't get an initial burst that was I lost so much of my drug with an initial burst that it wouldn't be a sustained release system. I would lose so much of the drug. So I said, visible light hydrogels, I'm getting rid of you. We're not gonna look at you anymore. Next. The next system that I looked at was, I said, oh, let's look at a different hydrogel. This was a thermoresponsive hydrogel that we knew from a previous project was injectable. And what's cool about this hydrogel is we were able to make it thermoresponsive. So at room temperature, it was sort of viscous and liquid-like, and it could be injected loaded into a syringe and injected into the eye. And then when it hit body temperature, it would collapse and allow drug to be released from it. I then loaded that hydrogel with tiny little nanoparticles filled with drug. And so then I said, well, I can manipulate the properties of the nanoparticles to get the release that I want, and then just put that in the hydrogel, and the drug can release and perform as I want it. Next. So this system was injectable. I was able to maintain bioactivity of the drug. I had a nice initial burst, but I still couldn't get the drug to release for greater than 10 days. Too much of it came out of the system too quickly, and so much of the drug was lost initially that I was concerned about the scalability of this project. I wasn't sure if I'd be able to make a lot bigger batches of these to work in a broader sense. And at this point in graduate school, I was several years into my project, and I was starting to get frustrated, and I was starting to think maybe this project wasn't going to work. Rarely in science do we have those aha moments where something suddenly works. But in a moment of frustration, I said, let me just put the vancomycin, put the drug directly into my thermoresponsive hydrogel. And I did. So I loaded in a bunch of drug and I tested it out, and it somehow miraculously worked. So it was a tight enough network to hold the drug in there, but not so tight that the drug wouldn't come out, and it actually released over the amount of time that I wanted, about 3 weeks. So here, next, on these slides, you can see this beautiful release profile of about 3 weeks of my drug releasing over time. I also needed to check to make sure that that drug release was bioactive, and so I did a little bioactivity testing where I took samples of the drug on each day, and I exposed it to the bacteria, and I made sure that I could kill the bacteria. And so what you're looking at here is the area around which bacteria has been killed from each sample, and they're all pretty good. It shows that they did in fact kill the bacteria. I then wanted to make sure that my system wouldn't cause any harm, and so I did cell cytotoxicity testing. I took samples from my system just filled with saline, so just looking at the system itself, and I exposed it to a bunch of cells, and the green cells are cells that are alive, and the red cells are dead. And I counted the green cells versus the red cells, and I compared it to controls. And next, all of the samples performed as well or better than the control. And so I maintained cell viability above 90% in all cases, suggesting that there was no risk of the system itself causing harm to the eye. The next thing I was looking at was degradability. And so I tested out to see if I could get these systems to degrade. And so I added in some additional chemicals, and what I found was all of the different hydrogels lost about 40% of their original weight, suggesting that they were degrading over time, but it was very, very slow degradation, which for the most part was a good thing because it meant that the hydrogel would not degrade so quickly that the drug would all come out all at once. Next. And so this system had pretty good encapsulation efficiency. I was able to get a lot of drug into it. I was able to get the drug to release for greater than 10 days. It was injectable, nice initial burst. It was scalable. And the one thing that I was missing was that the degradation wasn't quite in the time frame that I wanted. But I said, for now, this is good enough. This will work. My next aim was that I wanted to test this out in a rodent model. And so I got a bunch of rats, and I came up with 4 test groups. Go ahead next. So I had one group that received no treatment. All of my animals received an intravitreal injection of the bacteria cells. One of my groups received no treatment, so they just had a saline injection of saline just to see a control. Another group received a subconjunctival injection of the hydrogel without any antibiotics in it. My main test group received the complete drug delivery system loaded with a clinical dose of vancomycin. And my fourth group received a subconjunctival injection of the antibiotic as a bolus injection. And so that's what you would typically see clinically. I looked at 40 animals total, so 15 per group, and I had an experimental power of 75. And if you're not familiar with the idea of experimental power, this isn't a statistical test that you can do prior to an animal study or a human study that tells you how meaningful your results will be. And so if you're familiar with statistics, you may have heard of an alpha value. This is that beta value that you sometimes hear about, and you want to have a meaningful experimental power. And so it's good to do a power analysis before you come up with how many animals you need to do. Go ahead next. This is a nice little, previous. Go to the last slide. Yeah, this is just showing you what those gels look like. So in figure A here, you can see a blank gel. Figure D shows you a gel loaded up with drug. So it had a little color to it. And you can see there that it's injectable through a needle. And then over in figure C, what you see is that at body temperature, so if I held this hydrogel in my hand for a while and let it warm up, it starts to become opaque. And that's because it's starting to collapse. The network is changing and it becomes collapsed, which would allow drug to start diffusing out. Okay, next. So as I said, I was injecting these into the animal eyes. I won't go into a lot of details for how we did this, but we put the animals to sleep before we did it. They received their injections. We measured their ocular pressure to see if that changed. And we did each of the animals, each of their eyes. So the right eye of each animal received a control injection and the left eye received whichever treatment for whichever group they were in. Next. And so then what I did was I looked at each animal's eye and I scored it for how severely infected it was. So I looked at the cornea, the conjunctiva, and the vitreous, and I graded each of those on a scale from 0, which meant there was no infection, to 3, which meant there was a very severe infection. And then I sacrificed the animals at 12, 24, 48, and 72 hours post-infection and did histological examination of the tissue to look at what cells were present. And then I compared all to untreated animals. Next. So you can look here to see a little bit about what that scoring meant. So a normal score, an eye with no infection whose score is 0, has a very healthy looking eye that you can see everything, all the structures are intact, everything is clear. And as the scores get increasingly high, the cloudiness increases, there's inflammation, and you can see how bad the infection is developing. Next. Okay, so here are some of the eyes that I looked at, some example eyes. What you can see here is that at 12 hours, there was essentially no infection development. We couldn't see any infection. By 24 hours, though, the groups that did not receive treatment, so the group that received a saline injection or the blank drug delivery system without any antibiotic, were severely infected. Do you see how cloudy that cornea is and how there's redness starting to develop? And this persisted for 24 hours, and for 72 hours. The animals had severe eye infections. The animals that received treatment looked a lot better, and it seemed that there was no infection developing at all. And so if you look at the scoring, next, you can see that at 24 hours, there is actually no negative score, no infection score for the drug delivery system with the complete dose. There is still a little bit of a score for the group that received that bolus injection. By 48 hours, it's mostly the saline and the blank groups that have infections persisting, and this is true at 72 hours as well. And there's a little bit of inflammation, a little bit of score present for both the bolus injection and the drug delivery system. And here is that data presented in another way. So you can see that at 12 hours, there was no infection for any of the groups. At 24 hours, the bolus or the drug delivery system group still had no infection. But by 48 and 72 hours, there was a little bit of something, but we're still significantly less infected than the true control groups. Go ahead, next. You can also see this in the tissue. So these are the histology samples excuse me, for each of the groups. And what you're looking at here are some of the inflammatory cells that are hallmark of an infection. And you can see in the groups that did not receive treatment, so the blank injection and the saline injection, you see a lot of disorganized tissue, a lot of disrupted tissue. This is even true with the bolus group, but with the complete drug delivery system group, you see organized tissue without a lot of inflammatory cells, suggesting that there was no infection present. Next. And so what we saw at this point was that when you induce endophthalmitis, it will persist for at least 72 hours in a rodent model, and that our drug delivery system can help prevent endophthalmitis significantly better than a bolus injection at 24 hours and about comparably at 48 and 72 hours. There were some limitations, however, because we still saw a little bit of a problem at 48 and 72 hours. Now, at this point, I was ready to graduate, and I presented my work to my committee, and I graduated. So there's me with my advisor, there's me on graduation day, and I joined the faculty at Hulman Institute of Technology, where I had completed my undergrad. So if you go next, there's a picture of me with my current colleagues at our own commencement that we go to every year for our students. But you might remember that I was still one aim short, right? So I still had a third aim to my project, which was to develop a model for my system. Now, I had done some work on this in graduate school, and I just wasn't quite happy with how far I got. And so I continued working on this, and I continue working on it today as a faculty member. So I have my own students working on this project, and I'm going to take a couple moments to talk about that. So my goal here was to develop a computational model that could predict the loading dose required to achieve a therapeutic drug concentration in the vitreous and compare the different drug delivery routes to get drug from wherever you approach to the vitreous. And so what I did next was I took that release from the hydrogel, from my successful drug delivery system, and I inverted it. And I said, this is now the input of drug from that drug delivery system into the eye. I set some thresholds. So I said, okay, well, we looked at that, the ability of each sample to kill the antibiotic, and we said, what is the least amount of drug that we need to keep a nice little zone of inhibition, a nice circle of cell death? And I said, all right, 0.007 milligrams per mil is our threshold for minimum dosing required. And we built some compartment models. So here we have our compartment model from the drug delivery system entering into the subconjunctival space. And the drug must travel from the sclera through the choroid. There's some loss to choroidal blood flow, to the retina, to the vitreous. And we're looking at drug concentration in the vitreous. And I was curious to see, okay, well, let's not only look at the subconjunctival space, let's look at all of the drug delivery modalities or routes. And so next, we also looked at the precorneal and surface area. We looked at the vitreous, injecting directly into the vitreous. We looked at the subretinal space, and ultimately the goal was to compare how each of these performed relative to one another. We did a huge literature review search and looked at rate constants through all of these layers of tissue, and we modeled how the drug performed over time. And what we see here is that when you model drug in the vitreous over time following each of these modalities, you are able to hit some therapeutic thresholds in all of the cases, and then you can see the decline over time. And you can see how various of these are able to maintain a therapeutic drug concentration for a longer time than others. And so the topical actually maintains for the longest period of time, and then the least amount of time is maintained because of intravitreal injection, because it peaks with an injection and declines over time. I had a student who also built a computational model in ComSol that modeled the eye, and we did the same modeling. I'm not going to go into detail about that as well. But I still wasn't quite happy, and the fact that the topical area maintained the drug to the best of all the other systems bothered me, because I knew that wasn't really how this performs in reality. And so I have a student working now to refine the model, and we're seeing much better results that predict much better how the drug will actually perform when injected into an animal model. And so this is my current research, is developing this compartmental model that we can look at 4 different drug delivery routes, and we're going to be able to compare a bolus injection versus a sustained release drug delivery system. So next. Oh, sorry, go two later. One more. Oh, next. Yeah. And so my current work is refining that model, comparing the bolus injection versus a sustained release system, and seeing if we can make predictions about how much drug we need to put into the system in the first place in order to get a meaningful amount of drug in the vitreous for a long period of time. So I want to pause here because I'm at about an hour, and I want to see if you have any questions about anything that I've talked about so far.
Hey Emma, can you listen me? Can you hear me?
Yes, I can hear you.
Oh, great. It was a great presentation. And also for me, I had a mixture of feelings here because also I had a feeling of emotional one. It was very interesting to see everything you got in your research. And also I've seen some of those photos. Dr. King Miller, Chris, you. Oh, it was amazing to listen you, to hear you talking about your experience, also sharing with us everything you've got as a researcher and as a student and as an advisor, because I also left a little bit about some images you showed to us, some phrases, the adversary I've seen your in your slideshow. Yeah, it was amazing. And thanks so much, because I think this is the true thing the guys are going to see in the real world. So it's very good to know we are going to face those obstacles sometimes. But also when we see your presentation, the results you got, it's also motivating, because now we can see that and say, oh my gosh, I'd like to be like Emily and work on the same concepts and make the science got in advance. So yeah, it was great. Thank you so much, Emma.
Oh, thank you.
And now I have some questions here. Let me just see. The first question we got, I'm going to translate it from Portuguese to English for you, okay? So I have to take just some seconds to read in Portuguese and then in English. Oh, this one is in English. Could you explain a bit more how the risks are decreased when injecting the drugs into the different areas of the eye? What causes the retinal detachment when it comes to the bolus?
Yes, that's a really good question. The risk of retinal detachment is typically when you see a lot of repeated injections into the intravitreal space. My understanding is it's technique based a little bit. So if the surgeon is like slightly beveled to an incorrect angle, it can just put weird pressure on the retina that causes it to delaminate a little bit from the back of the eye. And so you obviously don't want that to happen. A single intravitreal injection might be okay, but there's always that the very conservative surgeon who says, I don't want to risk this, and so they go into the subconjunctival space, which doesn't have that risk at all.
Oh, nice. Okay, thank you, Emma. For the next question here, this one is, it makes sense to Brazil, and you may find the difference in the States, so feel free to say how do you think is that in your market. But they are asking, how do you think we could use those concepts, that technology, and share it with the industry, with the practical for the physicians, how they can access your technology and your science in the real world?
Yes. Okay, this is a great question. There is definitely often a disconnect between academic research and clinical relevance, and I would say this is not ready to go to the clinic yet. There are sustained release drug delivery systems that are used in industry. The problem with this particular one, I would say, is the fact that there may be degradation over time. And how does that work in terms of the shelf life? So something I haven't figured out about this system is if this is sitting in saline on a shelf for a year, how is that going to impact the performance of the system? We haven't tested that. Alternatively, we may want to freeze dry it so there's no liquid involved, but then there's the question of how that affects the drug integrity. And so I would say this is not ready for the clinic yet. You bring up a really good point about some of the challenges of doing sustained release drug delivery systems, because these are constantly the challenges: the shelf life, and the long-term implications of how can we get this from the lab into the patient without completely requiring the surgeon to have to generate the system in the OR, because that is one suggestion that has been made is maybe the surgeon has to make the hydrogel in the lab so that it's fresh. So that's a really good question.
Oh, nice. Thank you. And the next question is quite similar with the first. And this one is, do you think in the States, when you are a researcher like you have your PhD and you applied a lot of efforts to having your experience, do you think the industry have these, like to have this kind of professional with this research experience in their environment? Or do you think it's separated? I mean, you have the researchers in one side of the area and the developers and professionals of the industry in the other side. Or do you think in the States you have intersection point to join everything in the same table and make this advance to the country?
Okay, I think that's a great question. So I think it depends on the research that's being done. And actually, eye research is a really hot area right now. There's a lot of research being done, and there's a lot of collaboration between academia and industry. And in industry, there's a lot of research being done. So the research and development is big for ocular drug delivery. And so I think there is a lot of great communication happening between researchers and companies who are looking to fund and develop these systems. I don't think that's true for some other projects. And I think one of the challenges in academia is you have people who are sitting in a lab doing research in very interesting areas, but in ways that is never going to be translated into a practical system that could be used. And so they're pursuing it for sort of the sake of knowledge, but it's never going to be meaningful to the public. And then get into the question of, is this something worth funding or not if we can never actually use it? But I think for eye stuff, there's a lot of communication between research and like industry and companies.
Oh, great. The next question is about the appliance of your research and the drug delivery system with the hydrogels for cancer area. Do you think we can use the same concepts for treating people with cancer?
Yeah, I do. I think some of the future of sustained release drug delivery systems will be to target tumor tumors in tricky areas. And so, you probably are familiar with the limitations of, let's say, chemotherapy, because you're giving this systemic dose of a really toxic drug that not only harms the tumor but harms the patient as well. But if we can inject a sustained release drug delivery system that is somehow able to target tumor cells alone, or can go to a specific area, you're not going to have that systemic effect of chemotherapy. And so I think it is potentially like the future of cancer therapy. Especially, I know there are researchers, I'm not familiar with this research, but people working on smart nanoparticles that can actually recognize tumor cells and go to the tumor to release the chemotherapeutics versus just giving a systemic dose. So I think that could be really the future of cancer therapy.
Oh, okay, Emma. For the next question, I think you have given the answer for this one too, but it's about genetics. Do you think with the CRISPR-Cas9 and advancing in the genetics and tissue engineering, we could join the concepts from genetics with the nanoparticles and drug delivery system to have a better adhesion of the drugs into the cells?
Yeah, I think so. And I'm not sure if this exactly answers your question, but the cool thing about the nanoparticles and the thing that I really like about them is you can load them with almost anything. I'm looking into doing a sabbatical in a year, and the lab that I'm going to join is actually trying to load nanoparticles with DNA. And so there's the question of condensing the DNA to get into the nanoparticles, but then they can treat various conditions of the retina using, the DNA that's been loaded into them. And so that's true for anything that you wanted to deliver to part of the body. If you can get it into the nanoparticle, it can help with the treatment.
Oh, okay. So let me just check here if you have more questions. I think we do not have more questions. Emma, so that's it. Thank you so much, because when I was seeing your presentation, I've seen some photos and images you have produced in your work. And for me, it was a work of art. That everything you have presented to us, oh my gosh, it was amazing. And I'm very happy for having this contact with you and keep in touch with you after our experience together in the IIT. I'm very happy for having you here, and I'm celebrating inside myself, because it's a very special moment also for me. Thank you so much for sharing your knowledge and your experience with those guys. We have a lot of students here. Just to let you know, we have 3 spaces, and here where I am, and this is the lab where I work. And also, I almost sleep here and live here because I spend a lot of time in this lab. And here I have some students. We have another space with more students, and also we are transmitting this presentation in internet. So if the guys want to access our talk by YouTube, they also can access it. So
very cool.
you achieved a big audience.
Wow, that's great. Thank you so much. I really appreciate you inviting me.
Oh, thank you so much. Have a very nice day. I hope you enjoy your days in the place you are now. And thank you so much, Emma. After this point, I'm going to send more message for you.
Thank you so much. Have a great evening.
Yes, have a nice one. See you. Bye-bye.
Bye.
Hugs for your children, and bye-bye.
Thank you. We'll be in touch.
Pessoal, vocês que continuam aqui comigo aqui agora, né? A gente vai liberar um link para que vocês possam acessar o QR Code e acessar o link para poder participar do sorteio. Então, a gente vai deixar alguns minutinhos aqui o link na tela, já está aparecendo aí para vocês o QR Code. Acessem o QR Code, vocês colocam o nome de vocês, eu vou dar 12 segundos e meio, brincadeira. Vou dar um pouquinho mais de tempo para que vocês possam acessar esse link aí. Coloquem o nome e depois de um instante a gente vai fazer o sorteio da palestra de hoje, tá? Ao longo da palestra, a gente teve a lista de chamada, né, como o Marcos colocou aqui, fazendo em vários momentos ali, chamando vocês para poder assinar a lista para quem quisesse equivalência. E também, só um aviso breve, a gente teve um probleminha com relação à chamada para a tradução, né, a participação da Emma, da Emily Dosmar, em português, com a tradução em português. Foi um problema técnico que a gente teve, mas gostaria de agradecer demais a Maia Abramovic. A Maia é uma aluna aqui da Engenharia Biomédica e fez esse trabalho de tradução pra gente. Essa foi a Maia. A Maia, muito obrigado, Maia. Obrigado de verdade. Ela se predispôs com muita garra aí para poder fazer a tradução, mas infelizmente foi um problema técnico, acontece. Só que aproveito para fazer a chamada para vocês, porque na quarta-feira a gente vai ter uma palestra junto com a Jennifer Ken Miller, que inclusive apareceu na apresentação da Emma. E a Jennifer foi a orientadora tanto da Jennifer quanto a minha orientadora quando eu participei do intercâmbio ali com elas. Ela vai falar sobre coisas relacionadas a Drug Delivery System, sobre implantes oculares. Então, quem não conseguiu pegar a palestra de hoje por conta da tradução, que a gente teve problema técnico, na quarta-feira vocês vão ter uma outra chance também de ver que vão ser temas relativamente próximos aí. Tá bom? Tempo dado, Marcos? Pessoal, então, para quem acessou o link, está acessado o link. Quem deu o seu nome, está dado o seu nome. E agora a gente vai fazer o sorteio e ver quem que vai ganhar esse prêmio do webinar. O pessoal está preparando aqui. Quem ainda não colocou o seu nome, respire fundo, coloque o seu link aí rapidinho que a gente já vai fazer o sorteio. Oi, Bruce. Agora o Marcos vai fazer o sorteio aqui pra gente. Ele vai sortear aqui os 7 números que deixaram o nome. E dos 7, o ganhador foi o número, qual o número que deu? Número 1. Vitor Garanhari? Vitor Garanhares. Vitor Garanhares, se você me escuta, você foi o ganhador. Ele pode retirar o brinde dele na lojinha da SEB? Isso, você pode passar na lojinha da SEB ali embaixo do prédio 5 e retirar o seu prêmio, tá bom? Pessoal, todo mundo que participou, todo mundo conseguiu compreender, né, a apresentação. Espero que vocês tenham curtido. Essa foi mais uma experiência de palestra internacional. A gente tem começado com essas palestras desde do ano anterior. No ano anterior foi uma palestra, agora vão ser 2. Então, volto a reforçar, quem não conseguiu acompanhar essa por conta da tradução, na quarta-feira a gente deve estar nativo aí de novo para vocês poderem ver uma palestra internacional com a Dr. Jennifer Kang Miller. E agora, na sequência, a gente vai ter aula magna com um para-atleta que vai compartilhar um pouco a experiência dele aí com a gente, tá bom? Meninos e meninas e a todos, muito obrigado pela presença de vocês. A gente continua se vendo aí durante a SEB. Aproveitem bastante e até lá. Tchau, tchau.